Hello and welcome to Anatomy and Physiology 2. My name is Andres Gomez and I will be your instructor for this course. We're going to start with the chapter in the blood. These are some of the objectives that you should focus on when you want to know what are the takeaways from this class. So as we're talking about blood, you should know their main components, their functions and characteristics. Blood is divided into two components, okay? Formed elements and plasma. So, make sure that you know what is the composition of plasma, what are the functions of plasma. And when we talk about formed elements, we talk about three components of that. And that is red blood cells, white blood cells, and we talk about cell fragments, which are platelets. So as we are going into each one of them, make sure you know the structure, the function of red blood cells. You should know how it is that red blood cells are made and how it is that they are recycled. Continuing in this discussion of formed elements, you should know what are the functions of white blood cells, what is their function, what do they do, and for platelets, you should know their structure and function. We'll also dive into some of the mechanisms that the body has to prevent blood loss through blood clotting, and we'll also talk about surface antigens in red blood cells for blood typing. Okay, so let's get started with the circulatory system. So the circulatory system main or fundamental function is to move blood from one end to the other. And it's able to accomplish this with the heart, the blood vessels, and the blood. We will focus on the blood itself, but just keep in mind that the heart is that muscular pump that is pressurizing the blood through blood vessels in order to move the contents of blood. It is in blood that you carry the nutrients, the hormones, everything that you need to deliver to the other bodies in order to function properly. So how do you describe blood? Well, blood is a specialized connective tissue and it contains cells, okay, red blood cells, white blood cells, also cell fragments, platelets, suspended in a fluid matrix, which is called plasma. Now, what is within blood? Respiratory gases such as oxygen and CO2. Remember that you breathe in oxygen and you exhale CO2, the waste. Nutrients, whenever you eat a meal, those nutrients of the breakdown of the proteins, they are absorbed through your GI system, they are delivered to blood and then taken throughout your body. There are hormones, endocrine organs delivering testosterone, cortisol throughout your body. It is delivered throughout your body through blood. Your immune system has white blood cells. These white blood cells are the main reason why we have the body defenses. And we also have waste products such as urea, such as I'm just thinking of urea right now. But there is a key concept, and that is that these nutrients, these components that are in blood, they don't transfer from one cell to the other directly. They have to go through a intermediary player, which is the interstitial fluid. So we're gonna picture some cells in an organ. We're gonna picture a capillary and we're gonna picture blood. So there is XY nutrient. We're gonna think of oxygen going into it. Now it will not go directly into the cell, but it will go through the interstitial fluid. Actually, let's go ahead and make that blue. Let's make the interstitial fluid blue. So that is what the interstitial fluid is, the fluid surrounding those cells. So as oxygen diffuses into this tissue, it will go through the interstitial fluid and then into the cell. In this case, the cell will process that oxygen and then will deliver CO2 and then it will go into blood. So the flow of materials go through the intermediary player, the interstitial fluid. This is important to understand as we will have to know where fluid is at and how it affects different disease processes in the future. So to summarize, what are the functions of blood? First, to transport dissolved substances, such as oxygen, carbon dioxide, metabolic waste. There are proteins within blood that help regulate pH. There is also ions that are important for the functioning of your body. There is the restriction of fluid losses at injury sites. This is through blood clotting and platelets. White blood cells play a huge role against toxins and pathogens. And lastly, you should know that the blood runs slightly higher than body temperature. And because of that, your blood is able to maintain certain areas of your body warmer or colder depending on where you're shunting the blood. So for instance, if you're in cold weather, the person right here, if they're in cold weather, the blood will shunt to the trunk because there will be vasoconstriction in the periphery, and that is because we want to keep the trunk warm. All right, the trunk and the head warm. This is one of the ways in which we can stabilize body temperature. So this is a summary of the characteristics of blood. Blood runs at about 100 degrees Fahrenheit, slightly higher than body temperature at around 98. This is 98.6. This is important because by shunting blood flow from one end to the other, you are able to dissipate blood flow and you are able to conserve temperature where you need it the most. High viscosity. This is a key concept that you should remember. Viscosity stands for resistance against flow. Now, if you were to picture water and honey is more viscous than water because when you let them move down a slide, honey will take longer to get to the bottom. Water will get that much faster. And if now you compare blood to water, you will see that blood blood is 5 times more viscous than water. Now you should also understand that blood is slightly alkaline. The pH scale for blood, the pH scale goes from 0 to 14. 7 is neutral, and you are in that range from 7 to 7.45. If your pH goes above that, we term this as an alkalotic state. Alkalosis or alkalotic. The patient will have alkalosis. It will be an alkalotic state. But if the pH goes below 7.35, we will call that acidosis. Your body has different mechanisms within to keep that pH within range, either be through buffer systems, proteins, but if you are retaining acids or you are consuming too much acid, the scale will go towards the acidotic state. Blood volume. Another key concept here to remember is that about 7% of your body weight is in kilograms. Okay, about 7% of your body weight in kilograms is blood. So in males, it's about 5 to 6 liters, a little bit higher than females, and that is because males on average are a little bit bigger than females. Now, what is blood? Blood, again, is made out of plasma and formed elements. So plasma is mostly water. It has dissolved proteins and other solutes within, such as ions, such as hormones. Now we have the cell fragments, and here we refer to red blood cells, white blood cells, and cell fragments, which are platelets. Actually, here, as we're discussing, let's remember that the formal name for red blood cells is erythrocytes. The formal name for white blood cells is leukocytes, and the formal name for platelets is thrombocytes. Thrombocytes. You'll see that they have cytes as a common word. This means cell. Cyte. Erythro means red. Leuco means white. And thrombo means clot. So this is paint a picture that what is whole blood. So whole blood means what is in your body running right now. Whole blood is divided into plasma and formed elements. So plasma is made out of 92% water, and about 7% out of those, out of the plasma content is proteins. Remember that those proteins are divided into three, mostly albumin, globulins, and fibrinogen. The most abundant of the three is albumin, okay? And you should think, when you think of plasma proteins, you should think of liver because they are made in the liver. We're going to revisit all these concepts as we go, but it's important for us to have repetition on when we encounter these concepts. So we have whole blood, which is what's running in your body. Now that can be divided into two sections: plasma and formed elements. Plasma is mostly water, and it has a lot of proteins within, which are these three right here, amongst others, but these are the most abundant. Now let's talk a little bit about formed elements. So remember how we talked about white blood cells, red blood cells, and platelets? Most of the formed elements is red blood cells. About 99% of cells with informed elements are red blood cells. So there is a test that is called a hematocrit. This is a test that is used in the lab for patients to evaluate the status of the red blood cell count. Now the hematocrit checks to see what is the percentage of formed elements in whole blood. So they put in a sample of blood to a test tube. They put it in a centrifuge, and they divide the different components through density. Now, because red blood cells are the most dense, they will go to the bottom of the test tube. And the plasma component will stay in the top, and these will be formed elements. Now, because the formed elements is 99.9% red blood cells, the finding of this hematocrit, this percentage, is alluding more so to the status of how many red blood cells you have. You should know that it's about 50-50%. It's about 50-50%. It would shift depending on the cycle of that person. Now, these red blood cells, if we get a hematocrit of about 45%, it will be about normal. 45-50%, it will be about normal. But again, keep in mind that when we're doing the clinical test hematocrit, we're checking for the percentage of formed elements, but it is mostly alluding to red blood cells because that is the most abundant element within formed elements. When you look at white blood cells, now we're going to go into a little bit deeper, and that is that we have different types of white blood cells. And I'm going to give you an acronym for you to remember, and that is never let monkeys eat bananas. And that is the neutrophils, leukocytes, monocytes, eosinophils, and basophils. These are different types of white blood cells. And this acronym also tells you from most abundant to least abundant. So these are more common and these are least common. Each one has different functions, and we'll dive in deeper into each one of them and what they do later on. And these are the platelets. So thus far, we've created this discussion about blood. We're trying to move nutrients, blood flow from one end to the other. Now, blood is made out of different types of elements. We talked about plasma being mostly water, having some proteins within, and they're very important for different processes. Now we're going to focus on formed elements. So remember, formed elements is mostly red blood cells, about 99.9% of them. All right. They help specifically to move respiratory gases, and that is oxygen and CO2. So you breathe in oxygen, and as you breathe in oxygen, those oxygen molecules will go into the capillaries, the interstitial fluid, and then go into blood. And as you exhale, there is an exchange of CO2. You are releasing the waste. All right, so that is the main function of those red blood cells. White blood cells, again, there is 5 types. Never let monkeys eat bananas. So that is the 5 types of white blood cells, and each one has a different role, but they are all involved within your immune system. Okay, so if you think of red blood cells, you should think of respiratory gases exchange. If you think of white blood cells, you should think of the immune system. There are 5 of them. And then the last formed element, which is a cell fragment, is platelets. They are involved in blood clotting. A few key concepts to remember here. When we are talking about large amounts of red blood cells, we talk about something that is called oocyst. Means large amounts. Pinia means little amounts. So if we were to connect both words together, erythrocytosis will mean a high count of red blood cells. What about if we say erythropenia? Will mean a low count of red blood cells. What about leukocytes? Leukocytosis. Leukocytosis will mean a high amount of leukocytes. A leukopenia will mean a low amount of white blood cells. There are different reasons why this may happen. If you have more testosterone in your body for males, you will have erythrocytosis because you will have an increased production of red blood cells. If you have a GI bleed, you will start losing red blood cells that will cause erythropenia. If you have an infection, the white blood cell count will increase. If you have an immune disorder and you're not producing as many white blood cells, then you will have leukopenia. And the same goes for platelets. Remember that the formal name for platelets is thrombocytes. So for thrombocytopenia will be little amounts, and thrombocytosis will mean large amounts of platelets. Large amounts of platelets will lead to more blood clotting, will lead to blood clots in your body. Lack of blood clots in your body or thrombocytopenia will lead to bleeding. So we are now talking about some of the characteristics of blood and the creation of these formed elements. Hemopoiesis is the process by which we produce formed elements. We have myeloid and lymphoid stem cells in the bone marrow. Myeloid is responsible for most of the formed elements, and lymphoid is responsible for lymphocytes. So most of the formed elements come from the myeloid portion of it. Okay. Now, fractionation. Right? What we're trying to learn here is the basic concepts to understand what it is that blood does and the role of how your body functions. So just to summarize, we have blood with multiple components within, and it has different characteristics that are used by the body to help it function. Now, fractionation is a process that we humans use to determine how many formed elements are there in whole blood. Remember how we were talking about whole blood being divided into plasma and formed elements? This is a process in which we grab the blood and we put it into a test tube and we put it into a centrifuge and we divide the plasma and the formed elements through density. So as we are putting force in the test tube, as we put force in the test tube, the most dense elements will go to the bottom of the tube, formed elements, and the least dense, the plasma, the proteins, will go to the top. They are about 50-50%, about 50-50%. But because the formed elements are 99.9% red blood cells, this formed elements component, or the hematocrit, is mostly a reference to how many red blood cells we have. Oftentimes the hematocrit is higher in males than it is in females, and that is because we have a higher production of red blood cells. Because of testosterone, for one, amongst others. Now, moving on to the other side of the coin, plasma. So what is plasma? It makes up about 46-63% of whole blood, so about 50-50. 90-92% of it is water. About 10-7% of it is solids such as proteins. And remember, we talk about 3 different types of proteins, which is albumin, we talk about globulins, and albumin works as a buffer system for pH. It also helps carry other molecules, a carrier. It also helps mainly to keep blood, fluid in blood, water holder. So in other words, it helps increase osmolarity. And we'll dive into it in a little bit, but this is one of the biggest players on maintaining the osmolarity of blood the way it is. And lastly, for the third type of a protein is fibrinogen. Fibrinogen is the precursor to fibrin, and this plays a role in blood clotting. Now, globulins play a role in immunity. They are also carrier molecules, but you should think of immunity. Another key concept here is the difference between interstitial fluid and plasma. The composition is very similar, but the difference between the two, the major difference between plasma and interstitial fluid, is plasma proteins. Because we have albumin, globulins, and they are non-permeable proteins that do not cross capillaries. They create an oncotic pressure, all right, that pull the water in, and with that you allow to keep water into blood, all right. So you should, another key concept you should remember is osmosis, which is the movement of water from one side to the other through a membrane. In this case will be the capillary. You should also know osmolarity. What is osmolarity? The concentration of solids in a solution. So the osmolarity of blood is dependent on those solids that are within, such as albumin, globulins, fibrinogens. And these plasma proteins are non-permeable. That means that they don't cross the capillaries. Diving a little bit more into plasma, when you think of plasma, you should think of, sorry, let me just put in here, liver. Made in the liver, made in China. No, they are made in the liver, about 90% by the liver. So whenever a patient has liver disease, you should think of a deficiency of these proteins. So if you have less albumin, that means that you have a lower osmotic pressure. So with that, you will not be able to pull the water back into blood. So the blood will will stay within the interstitial fluid. Let's just create a picture right here. These are a couple of cells. This is blood, and we have less of albumin. So we have a decrease in albumin. The water, let's go ahead and make it blue. The water will increase on this side, on the interstitial fluid. There will be a decrease to the flow of water will go in this direction. So blood pressure will decrease because there is less blood over here. The patient will swell because there will be edema. There will be more blood in the interstitial fluid. This is this key concept right here of osmotic pressure. It helps maintain. Large size prevents the proteins from closing. They are non-permissible. All right. Specific proteins, albumins, you should think that it helps you buffer acids. It helps you transport molecules. Globulins, they work as antibodies and they also help with transport. And fibrinogen is the precursor to fibrin, which plays a key role in blood clotting. So when you think of liver disease, think of patients that are swollen because they don't have enough albumin to bring back the fluid into blood. Now, formed elements. Within the red blood cells, you should know that the formal name is erythrocytes. Again, erythro means red, cytes means cell. That is the formal name. If you wanted to describe that there is a lot of them, you will say erythrocytosis. And if you want to say that there is a little of them, just a few of them or a few amount, erythropenia. But this will be in relation to the ranges of a normal person, right? Since we know the ranges what a normal person is at, if you are below that range or above that range, you will use these terms. Again, red blood cells make about 99% of formed elements. So when we do a hematocrit, even though it is the percentage of formed elements, it is more of a test to tell us how many red blood cells do we have. Why is blood red? Because of hemoglobin. Hemoglobin is red. It gives it that pigment to its color. And what is the major function of red blood cells? To carry respiratory gases, oxygen and CO2. You breathe in oxygen, you breathe out CO2. Red blood cells. So red blood cells are the small discs that are biconcave. So they are very flexible because the capillaries that they go through are just sometimes a fraction of what they really are. So they have to bend onto themselves to carry, to go through these capillary vessels. They have a biconcave shape that allows them to stack onto each other, okay, creating something that is called Rayleigh. We'll look at it in the other slide, but they're small discs. They are biconcave. They are very flexible, all right, and they contain hemoglobin. It's the hemoglobin that carries the oxygen, and because the main function of the red blood cell is to carry oxygen, most of the cell is hemoglobin. Now there are different stages to red blood cells. Just know that they mature mature red blood cell does not have a nucleus. Let's go ahead and go through the stages very briefly. You have erythrocyte colony forming units. These units get stimulated by something that is called EPO, erythropoietin, that is released by the kidney. So when you are lacking oxygen, a decrease on oxygen levels make the kidneys release EPO, and the EPO goes to the bone marrow, and the bone marrow this stimulates the erythrocyte colony forming unit, and then that leads to the development of erythroblast. You can remember that the first one is the first one is the first one because it has a B, and then you can remember that the baby red blood cell. So B stands for baby. So the erythroblasts at this point they still have a nucleus and they have all the organelles. It is here where they start creating all the hemoglobin, okay, and then they transition to something that is called a reticulocyte. Now, they haven't left the bone marrow yet, but when they cross over to a reticulocyte, the nucleus is destroyed. The nucleus is destroyed. It is mostly hemoglobin at that point. And then after that, then it will migrate to the periphery, and then it will mature. This process takes about 3 to 5 days. All right, and it is in the periphery that that cell now can live up to 120 days until it dies. It cannot live any longer because it doesn't have any organelles. It doesn't have a nucleus. It mostly has it doesn't have mitochondria. It only has a couple of mechanisms to maintain the cell the cell membrane, right? It has to be very flexible. All right, we'll dive into this a little bit deeper, but this was just a brief introduction. Okay, so red blood cells, remember that they are very small, they are very flexible, they are biconcave, they have a high surface to volume ratio, and this allows them to quickly absorb and release oxygen. They can stack onto each other, and when they stack onto each other, they form something that is called the rouleau. Now, this allows them to go through narrow vessels. Because of this shape and the stacking form, they are also very smooth. The movement of red blood cells is very smooth, even through very small vessels. It is important for the movement of blood to be smooth because if it is not, it may cause blood clotting. This is alluding to the fact that they are going through places that sometimes it is smaller. So this is the size, and sometimes the capillary is at 4 micromilliliters, so it has to fold onto itself. So this is a smear of whole blood. You will see that most of it are red blood cells, and this is what they will look like in a three-dimensional picture. This is a capillary, and you can see how here they're stacking onto a rouleau here, and these are blood vessels. Okay, so the lifespan of a red blood cell is about 120 days. The formation of a red blood cell takes 3 to 5 days. Now it is the reticulocyte and it is the mature red blood cell that is missing nuclei, mitochondria, and ribosomes. The reason why it doesn't have mitochondria is because we're trying to transport oxygen, and the mitochondria uses oxygen for cellular respiration. So if it had mitochondria, it will be consuming the oxygen that it's supposed to carry to other tissues. It is the red blood cells, the only cells in the body that work through anaerobic respiration. And they like ribosomes because they're not creating new proteins. It cannot perform repairs. It cannot make proteins, so the energy demands are low. So that is why anaerobic cellular respiration is the only form that they have to repair the cell membrane. They like mitochondria again because they will be consuming the oxygen they are supposed to deliver to other tissues, so that wouldn't be good. Okay, so we talked about red blood cells, what they look like, how flexible they are. We talked briefly about, how it is that they are made. Now, what is the main component within red blood cells that helps them carry oxygen? And that is hemoglobin. Hemoglobin is a quaternary protein, and that is a complex protein, and it's made out of 4 globulins. All right, within the globulins, I want you to know that there are three: alpha, beta, and gamma. I believe that it's like that. So alpha, beta, and gamma. So hemoglobin is made out of two alpha chains and two beta chains, at least for adult hemoglobin. The hemoglobin for adults and in fetuses is different. So just know that overall the hemoglobin is still four chains of something. They both have alphas, but for fetuses, so fetal hemoglobin is two alphas and two gammas. Show you. Okay, and two gammas. And this one has a higher affinity for oxygen and allows it to obtain the oxygen from the mother. This is a concept that we'll revisit later on, but just know that hemoglobin is made out of, four quaternary proteins, and they are globulins. They carry within something that is called a heme group. And it is within this heme group that you can carry oxygen. Each hemoglobin molecule will look like this, will carry one molecule of oxygen. So in the center right here, there will be that heme that we talked about. The oxygen will be right here, right here, right here, right here, and you will carry oxygen, an O2 molecule right here, O2 molecule. So you will carry four molecules per hemoglobin. All right, and the center right here is because there is an iron right here. It's the iron that helps with that. Okay, so hemoglobin, we talked about, let me just go ahead and put this in here. It's a protein molecule that transports respiratory gases. Where is the respiratory gas? The oxygen, and is through that heme. CO2, the other respiratory gas, travels more so in the periphery. Okay, travels in the periphery. It doesn't go into the heme group. There are multiple red blood cells. There are millions. Okay, now each hemoglobin molecule can bind into four molecules of oxygen and four molecules of carbon dioxide, but it will be on the periphery. So just to recap, hemoglobin is a complex quaternary structure made out of four globular protein subunits. So four globular protein subunits. Each one of those proteins has a heme group that carries iron, and it is to that iron that the oxygen binds to, and that is how we describe oxyhemoglobin and deoxyhemoglobin. So when it associates, it is oxyhemoglobin, and when it is not, it is deoxyhemoglobin. The iron within the heme group is the one that, let's go ahead and zoom it in. Av2 plus O2 plus. So you would think that oxygen normally it will normally create a reaction with iron, but there is a balance back and forth, so it doesn't completely oxidize iron. It stays in a balance and is able to absorb it and release it when it needs to. Now it is in the periphery that the CO2 will be bound to. Okay, not within the heme group. This is your heme group, and this is one of the globulin proteins. Okay, so this is another picture for the oxygen binding to your heme group. This is your alpha chains and your beta chains. These will be adult hemoglobin. So rehearsing some of the functions of red blood cells, remember that the main function of red blood cells is to carry respiratory gases. They will pick up the oxygen in the lungs and will release a carbon dioxide. Now this oxygen that is picked up in the lungs is distributed to the periphery, and it's in the periphery that it will pick up that carbon dioxide and bring it back to the lungs. So in the lungs, you release the carbon dioxide and you bind oxygen, all right? And it is from the lungs that you start bringing to the periphery that rich oxygen blood, and then you will deliver that oxygen, and then you will pick up the carbon dioxide. Now, why is oxygen important? Oxygen is important because it is used in cellular respiration. Your body will need ATP production, and we use this in mitochondria. So mitochondria will use the oxygen to produce ATP through cellular respiration. Now the byproduct from that cellular respiration will be CO2, which is the excess gas, and that excess gas will go into that red blood cell. So now that we know how red blood cells are made, let's talk about how they are recycled. About 1% of red blood cells wear out and are replaced every day. And that is again because they don't have organelles and they have to continuously bend and fold when they go through a small a capillary, they might just break. Macrophages are the main players at players here because they help recycle red blood cells. They engulf worn red blood cells and they lyse them, they break them, and they try to separate some of the globulins, some of the heme groups, some of the iron, and they deliver it to the different places that is needed. Now macrophages in the liver, in the spleen, and the bone marrow begin that recycling. Macrophages, I want you to think that it's like a huge cell that engulfs a red blood cell. And inside there, it can start that process of breaking down the red blood cell, and all the components out of it start taking them to the right places. You do not want to, or it's not ideal for the red blood cell to break outside in blood because it will release all these components in blood and they can be toxic. All right. So these macrophages, in the liver, in the spleen, the bone marrow, they are monitoring those red blood cells and they hemolyze them when they are not good anymore. And they remove the hemoglobin molecules and iron after hemolyzed. So continuing on this discussion of the recycling of red blood cells, the macrophage is engulfing that red blood cell and is hemolyzing it, breaking it down. Now, because the red blood cells are mostly hemoglobin, the hemoglobin will be broken down, okay, because it's mostly protein, into amino acids, okay. Now remember the heme group that carries the iron and the oxygen. The heme group is also processed, all right. Here, the heme group within the macrophage is converted to biliverdin. Now, biliverdin is still within the macrophage, is then converted to bilirubin. Bilirubin is not as toxic anymore and is released into the bloodstream. And while it is flowing into the bloodstream, then it is filtered later on for waste. We'll get back to this component in a little bit, but just know that the heme group is converted to biliverdin, and then the biliverdin is converted to bilirubin. And the colors of bilirubin is yellow, biliverdin is green, and it's bilirubin that ends up being in the blood. The globular proteins are converted into amino acids, and the iron is released into the bloodstream. Now, iron oftentimes doesn't travel by itself. It needs a molecule to help him travel around, and that is transferrin. Okay, which brings it to the bone marrow. Transferrin also picks it up from the stomach. So when you eat and you have a meal and there is a iron-rich meal, gastroferrin will bring it into the bloodstream, and in the bloodstream, transferrin brings it to the bone marrow, and it is reused to make new hemoglobin. So bilirubin, remember that bilirubin is yellow, and it is the end product after the macrophage is done processing that heme. And then as it is in blood, it is then filtered out by the liver. The liver filters out the bilirubin and it is used to then be concentrated into bile. All right. Now there is a couple of steps in between. The bilirubin is converted into urobilins, and enterocobilins in the large intestine. This is then eliminated as feces and urine. So bilirubin is filtered mostly by the liver, all right? Now, because it is mostly filtered by the liver, if a patient has liver failure, you should think of a buildup of bilirubin because there is nothing to clear it out. And because the color of bilirubin is yellow, most patients will look jaundice. Jaundice is a sign that there is a buildup of bilirubin, either because of the liver not working, inflammatory factors, or because there is an over count of hemolysis. The red blood cells are being destroyed, either by an infection or because of X or Y reason. There is an over hemolysis of red blood cells that will lead to an overproduction or the recycling of the heme groups by the macrophages that the liver cannot keep up with. Um, Now, you should think of the fetuses also. Fetuses, because they have immature livers, they will naturally have some bilirubin buildup. We could address this, or sunlight helps break down that buildup of bilirubin. And here we have associated with mature, immature, or failing liver. Okay, so immature refers to an immature liver or failing liver. Focusing a little bit more into the pathophysiology and red blood cells, another type of disease that is seen within the human body is hemoglobinuria, which means that there is hemoglobin within the urine. This may make the urine appear red, not because there is blood, but because there is hemoglobin. When there is a rapid lysis of red blood cells, it will lead to an overabundance of hemoglobin in blood, and because blood is being filtered by the kidneys, this hemoglobin will pass through the kidneys. This could be a sign that there is a breakdown of red blood cells. Now, hematuria is speaking not to the buildup of hemoglobin in the urine, but red blood cells, actual red blood cells. Now, there is a couple of reasons why red blood cells may be in urine. Now, it could be due to injury, either via the kidney, later on in the kidney, in the bladder. However, know the difference. Hemoglobinuria refers to a hemoglobin in the urine, and it may make appear the urine red. And hematuria means that there are red blood cells in the urine, and the urine can also appear red. There are multiple reasons why they may occur. This is a little bit on to that cycle of iron formation and red blood cells. So this is, we're gonna start with the macrophage. The macrophage is engulfing this red blood cell and is breaking down the hemoglobin into amino acids, and is breaking your heme into biliverdin and then bilirubin, and then it's released into the bloodstream. And it is in the bloodstream that the liver will pick up the bilirubin and it will secrete it into the bile. Okay, and then the bile is the bilirubin then is converted to urobilins and enterocobilins. This part you don't need to know. However, as we move forward into this concept, we get down the iron after breaking down that red blood cell, and we release it into the bloodstream. And the iron is then carried by transferring to the bone marrow. And in the bone marrow, there is that 3 to 5 day process with erythroblast, reticulocytes, and mature red blood cells. I'm just doing abbreviations. But once they are done, the red blood cells will go on to live for about 120 days, transporting oxygen, and then they go. This production of red blood cells, remember that kidney releases something that is called EPO in the presence of hypoxemia. When the kidney doesn't have oxygen, so low levels of oxygen will lead to hypoxemia, leading to an increase on EPO production. Red blood cells. Remember that red blood cells go through erythropoiesis, or red blood cell formation, Erythro means red, poiesis means formation, and this formation happens at the red bone marrow. All right. We have hematocytoblast. These are the stem cells. Another word for this is hemo. We're gonna call it hemopoietic stem cell, and which is basically this one right here, the hemocytoblast. So this is another name that is used, hemopoietic stem cell, and it is from the hemopoietic stem cell that we will go into a colony for main unit. Actually, the difference is erythrocyte colony forming unit. So ECFU. So erythroid colony forming unit. These ones receive EPO stimulation, and then they decide to divide further into, let's go ahead and change it to black, erythroblast. And you know that these are still in the bone marrow because they are B, this stands for baby. And then for erythroblasts, they will start creating that hemoglobin, and once they're done with that hemoglobin, they destroy the nucleus, and then they become reticulocytes. And then when the reticulocytes are done producing, then they will go into actual mature red blood cells. Actually, the book doesn't mention as mature red blood cells, they just say red blood cells. Okay. So in this terminology right here, just remember that we have stem cells. They divide to produce myeloid stem cells and lymphoid stem cells. The lymphoid stem cells give rise to lymphocytes, but it is the myeloid stem cell that gives rise to all the other ones. Here we have the myeloid stem cell give rise to the erythroblast, and then the reticulocyte, and then the mature red blood cell. Here we're still in the bone marrow, and here we are in circulation. So we have the red bone marrow. So there are different names here. I want you to know that there is a myeloid stem cell, which is the hemopoietic stem cell, and then that can go into a colony forming unit, and within the colony forming unit, then you create the erythroblast. There are different stages of the erythroblast, but the idea is that once you are done creating the hemoglobin, then you will go into a reticulocyte. That reticulocyte will not have a brain, or sorry, a nucleus. So the erythroblast has a nucleus because it's producing hemoglobin, and the reticulocyte doesn't have a nucleus. All right, and they mature, but it has some ribosomes because it's still producing some proteins, and then at the end of it, it gets rid of ribosomes, mitochondria, and nucleus. And by the time that it is mature, it doesn't have any of it. Red blood cells. Now, remember that red blood cells production is expensive. So that means that we need to have a lot of nutrients. We need to have amino acids. We need to have iron. We need to have B12, B6, folic acid. So all of these nutrients are required for red blood cell production. So if we don't have iron, well, we cannot produce red blood cells. It doesn't matter how much we need them. Well, the quality of the production also will come into play. If there is not enough iron, the cells will start looking small. And if we don't have enough B12, the cells will start, showing large. So we need to have the right amount of each one of these nutrients in order to create red blood cells. Again, we're coming back to this molecule, which is EPO. It is produced by the kidneys, and the name of this EPO hormone is erythropoiesis stimulating hormone. It's secreted by the kidneys when there is no oxygen. So for instance, if you go running and you sprint, there will be a lack of oxygen in your tissues. They will lead into hypoxia. This exercise and strenuous hypoxia state will lead to your kidneys to send a signal, EPO, to the bone marrow to, hey, look, we need more red blood cells. Remember that there is erythroblasts that have a nucleus, and it will start increasing hemoglobin synthesis and red blood cell production. Something to keep in mind here is that if you see that there is a high count of erythroblasts, it means that there is an increase in production of red blood cells, either B because the person is bleeding, or B because they have EPO. Okay, either B because they're doping, so professionals, athletes sometimes may use EPO to increase their oxygenation status by increasing the amount of red blood cells. This is not all gain, because if you increase your red blood cell count, that also means that you are at a higher risk for blood clots, because they need to be released. Red blood cells have to have a space to move, but if you have more, it is more likely that your blood will be turbulent, will be more viscous, and with that, it may clot easier. So erythroblast is just the baby cells, remember, just for the nucleus. During anemia, you may find that red blood cells is decreased, but in the short term, depending on the type of anemia that you have, but in the short term, you may find that the erythroblasts will increase, and then long term, they will decrease. Decreased blood flow to the kidneys, hypoxemia, because if there is less blood flow to the oxygen, that means that there is less oxygen delivery to those tissues, will cause the kidneys to release EPO. If the oxygen from the lungs decreases, such as in high altitude, there is less, for every breath that you take, there is less concentration of oxygen. That will lead to a lower concentration of oxygen in your body. Again, decreased blood flow to the kidney, or the same blood flow to the kidneys but with lower oxygen contents, will lead to a production of EPO. We're almost coming into an end of this lecture, and we'll finish the remainder of the slide deck on a different video. So this is just summarizing some of the different tests there are that we can do clinically to evaluate a patient. Hematocrit, remember that it is the percentage of formed elements in whole blood, and the percentage of formed elements in whole blood is mostly red blood cells, and it is somewhere about 50%. Okay. Polycythemia is a term that is used to reflect high counts of, actually, let me just erase that a little bit, high counts of red blood cells. Okay, we have here erythrocytosis or leukocytosis. This is to express high amounts of red blood cells or high amounts of white blood cells. Reticulocyte count will tell you if there is a high turnover of red blood cells. If you have a high reticulocyte count, it means that there is more production of red blood cells. Remember that the production of red blood cells takes about 3 to 5 days. So in 3 to 5 days, if you start seeing this, it means that there was an incident about a day, 2 days ago that led to an increase in reticulocytes. Reticulocytosis then will mean that you have a high amount of reticulocytes, and it means that there was some form of bleeding. Hemoglobin concentration will, it refers to how much hemoglobin there is in your blood. If it is low, it means that there is a decreased amount of red blood cells. Red blood cell count, this is measured in million, while white blood cells is measured in thousands. Here we're talking about 4.2 to 6.3 millions per microliter. All right, so erythrocytosis or polycythemia will talk about elevations in red blood cells. Now, the results, if they are lower, it will mean that there is anemia. If there is a decrease in the hemoglobin, it means that there is anemia. If there is a decrease in your hematocrit, it means that there is anemia, which means that there is not enough red blood cells. MCV talks about the size of the red blood cell. So when we don't have enough iron, then your cell won't be produced as well. So we will have microcytic anemia, right? Microcytic. Macrocytic, it is if you're lacking B12. So lack of B12, lack of iron. All right. Mean corpuscular hemoglobin concentration. this is talking about whether or not there is enough hemoglobin within. So if there isn't enough hemoglobin within the red blood cell, if there is not enough, then it won't be as red. If there is a lot of hemoglobin, it will be hyperchromic. All right. And then lastly here, just remember that red blood cells are a circular biconcave shapes. They're very flexible. They don't have organelles. And they are used to transport oxygen and carbon dioxide to and from the lungs. And they remain in bloodstream for about 120 days. Thank you and see you next time.